Hello, my name is Ayan Mandal. I'm a second-year medical student at the University of Pennsylvania, and I recently completed my neurosurgery rotation. Before medical school, I did a PhD at the University of Cambridge, and I thought I would present some of the results of my PhD to the department, as I think it will be of considerable interest to a neurosurgical audience. My talk is titled "On the Origins of Glioma: Insights from Brain Network Mapping." So over the past couple of weeks, I've had the privilege to participate in a range of surgeries to resect diffuse gliomas from the brain. Despite the fact that we provide excellent care for our patients, it's staggering the fact that this disease still remains incurable. So why is that the case? I think a metaphor from Alice in Wonderland is quite helpful in elucidating this. This is called the Red Queen idea. So this is a character who has to constantly run and run and run just to keep in the same place. And I think our cancers are kind of like that too, where they're constantly evolving, and our therapies have a hard time keeping up with the rapid progression of this disease. Now, already our approach to gliomas is multifactorial in nature. We perform surgeries, we offer radiation therapy. In certain circumstances, we also provide chemotherapy. But despite our best efforts, the cancer still escapes our treatments and tends to spread elsewhere. So in my research, I'm very much interested in how we can better understand where these tumors are coming from and how they're spreading around in the brain. Hopefully, if we understand the biology of this type of behavior, we'll be able to better target it with therapies and maybe slow down this cancer so that our other therapies can catch up. So I'm presenting this now so that you know where I'm going with all this. Our general hypothesis for why tumors are where tumors are coming from and why they're going where they're going is that these tumors are originating from the subventricular zone and spreading along brain networks. We think this based on research from many other groups and also some of the results I'm going to show you today. Hopefully I'll convince you of this hypothesis, and we'll talk in the end about the limitations of it, but this is what we think the model is, and we think it's going to have important implications for how we think about new therapies to target diffuse gliomas. So here's our roadmap for elucidating glioma spread. To better understand this phenomenon, where we're going to start is by better understanding how gliomas are interacting with their neural environment. So we think a lot about the immune microenvironment of gliomas. There's also a neural microenvironment. Gliomas are interacting with the healthy brain or with functioning neurons. And there's been some really exciting work from Michel Mongier and colleagues and others looking at how glioma cells are forming synapses with neurons and how neuronal firing can sometimes influence the progression of gliomas to become malignant and spread elsewhere in the brain. We're interested. The work I highlight here is looking at the microenvironment, and in my work, we're really interested in the macro environment. So how are gliomas interacting with large-scale networks that we know are also involved, that could also be influencing the development of these tumors to spread distantly. And where we think this is interesting because we're oftentimes measuring the progression of gliomas, and clinically at least through neuroimaging. So it's an interesting question, an important question to address. What role are these more macro scale networks playing in influencing the development of these diffuse tumors? So the first study I'm going to talk to you about is one where we're taking advantage of another study that we did that was involved in testing intraoperative ECOG for monitoring executive function in patients, hopefully to better plan surgeries. But in this particular study, we took advantage of that, of the fact that some of the electrodes that we placed were placed on tumor-infiltrated tissue itself. And our question here was, to what extent does tumor-infiltrated tissue participate in large-scale cognitive circuits? And so how we went about testing that is we had patients intraoperatively as we're using electrode strips to monitor neural activity, we're testing them during three conditions. One, baseline at rest, where they're not doing anything. One, where they're simply counting from 1 to 2 to 3 and so on. And another where they're switching between 1 and A and 2 and B. And so because this is a more complicated task and this is an easier task, we can compare the electrophysiological signal between those two conditions and come up with a measure of cognitive activity that we can map onto the brain. And specifically, we're interested in whether we could detect that signature in tumor-infiltrated tissue, A, and B, do those task-responsive regions of the brain, to what extent are they interacting with large-scale connectivity networks that we know are associated with executive function and health? And I'll talk a bit more about that a little bit later. So first, just looking at how these results map onto the brain. So what we're doing here, again, is we're taking the electrophysiological signal during these different tasks and comparing it to one another. Specifically, we're looking at high gamma power, which is our frequency band that's most strongly correlated with neuronal activity. And we're seeing how that hard greater than easy contrast maps onto the brain. And we find for each patient, there's significant cognitive response in tumor-infiltrated tissue. So you can see that here with patient 1, you can see a significant response with patient 2, patient 3, patient 4, all in tumor-infiltrated cortex, which is represented as these grayed out regions corresponding to where the tumor is. So the prior results, this we expected to some extent, since we know that the tumors are infiltrating the brain diffusely and preserve function in many cases. Here we're interested in the connectivity of tumor-infiltrated tissue using bold fMRI. So what that means is we're taking voxels within the tumor, sort of represented here in red. That corresponds to where we found the task-responsive electrodes. We're asking to what extent is each voxel in the brain correlated with with the signal in tumor. And you might expect maps of connectivity to tumor to be random, but we found that, like in each case, we derived connectivity maps which were meaningful and interesting to interpret. So what we have here, so the map itself is colored from blue to red, red representing high connectivity, blue representing no connectivity. And the topography of this map follows these boundaries drawn in dark red, which correspond to canonical networks that have been identified in healthy individuals. And these networks often correspond to executive function and are important for executive function and health. So we find that the networks maintain their connectivity with the distributed brain regions involved with executive function, even after tumor has infiltrated those regions. That's the takeaway here. And finally, we're interested in statistically testing the correspondence between our measure of executive function from ECOG on the one hand, and that represented by percent signal change with a hard greater than easy or easy greater than rest tasks, and functional network connectivity from fMRI on the y-axis. We find especially a concordance when we look at the dorsal attention network, which is a network associated with goal-directed attention and high order executive function. And we find a significant association there, establishing a concordance between fMRI and ECOG in terms of identifying functional tumor-infiltrated tissue. So our main takeaway here is that diffuse gliomas are not only integrating with their local brain circuits and local environment, they're also integrating with these large-scale brain circuits. And that begs the question of what role might these networks be playing in terms of the progression of the disease and vulnerability of different regions to gliomas? Are these networks making a difference in terms of the biology of this condition? And so that's what we did next. And specifically, we're interested in this question of why gliomas appear where they do in the brain. And we published this work in the journal Brain. here we're interested in the fact that gliomas are not distributed randomly throughout the brain. So they tend to prefer certain brain regions. For example, you almost never see gliomas, or diffuse gliomas at least, in the occipital lobe, but you see them quite commonly in the insula and quite commonly in the frontal lobes. The question is, like, why do we see the distribution that we do? And our first hypothesis for explaining this distribution is that maybe these tumors are appearing near brain hubs. Hubs referring to especially highly connected regions of the brain, which many studies have shown that neurological diseases in general tend to like brain network hubs. I'll talk a bit more about what that means in just a minute. But first, I want to go over how exactly we derive this map of where gliomas appear most frequently. We're taking advantage of this global data set called BRATS, which collected glioma scans and lesion masks of gliomas from all across the world. We're taking those masks and mapping them onto a common template and concatenating those masks to define this map of glioma frequency. Once we have that map, we're interested in knowing the relationship between it and other variables of interest in the brain. First, starting with brain connectivity and brain network hubs. So as you all know, the brain is a richly interconnected network, as you can see from this image from the Human Connectome project. What we can do is we can take these like fancy animated maps like this and turn it into something more workable by representing it as a graph, which is a mathematical object composed of nodes, which are these like circles here, and edges, which are connections between the nodes. And in our particular case, we're representing brain regions as these nodes, and our edges are connections between different regions of the brain. Now, once we have a mathematical object like that, we can segregate these regions into things being hubs versus non-hubs. Hubs being regions that are especially important for communication across large-scale brain regions. And we know from prior research that these hubs are metabolically expensive to maintain and can be expected to be especially vulnerable to neurological diseases, including gliomas. And so if we're, here's a simple sort of binary way of thinking about hubs as more connected versus less connected than the average. We can take that measure and plot it onto the brain and make it continuous as opposed to binary. And we end up with a map of hubness, sort of shown here in red and blue. And when we take that map and look for the correspondence between that and our map of glioma frequency, we can appreciate a pretty strong correspondence statistically and also visually between this map of hubness and glioma frequency. So that was our first hypothesis. Second hypothesis, and by the way, these are all non-mutually exclusive. We know that there are possible cells of origin of glioma that are non-randomly distributed in the brain, and maybe the locations of those cells of origin can help us explain why tumors are showing up where they are. And so in pursuit of this hypothesis, we first started with the idea that gliomas might be more frequent in regions populated by neural stem cells. So neural stem cells are active and present in the adult human brain. This is a recent finding, but we know that they do exist in the dentate gyrus of the hippocampus and the subventricular zone. And so we took masks of those regions and compared them to other regions of the brain to see if there is a statistical effect of gliomas being more frequent in these regions enriched with neural stem cells, and we find that to be the case. Second, there's also this idea that gliomas might be coming from oligodendrocyte precursor cells. We derived a map of oligodendrocyte precursor cell distribution throughout the brain. And we found that that map also significantly correlated with our map of glioma frequency to a pretty high extent. So finally we're interested in this genetic hypothesis, the idea that there are certain genes that we know are very vital for gliomagenesis, and is it the case that where those genes tend to be expressed in the brain are where we tend to see gliomas most frequently? So to test that hypothesis, we made use of this atlas of human brain gene expression from the Allen Institute for Brain Science. We took their map of gene expression and co-registered it with our map of glioma frequency so that we can test which genes have spatial transcription patterns that best match our map of glioma frequency. And when we do that, we find two different categories of genes which correlate significantly with glioma frequency. One category is of genes involved in processes like chromatin organization, protein localization to microtubules, cell cycle, G0 to G1 transition. So genes that we know are involved in the cell cycle and gene expression, and one sort of category here. And a second category that's more so enriched with genes involved in neuronal signaling factors. So things like regulation of glutamate receptor signaling pathways and behavior and cellular metabolism. So we have two different categories of genes that appear to have spatial transcription patterns that match our glioma frequency map. And we found that this first one here is significantly enriched with genes that we know are related to gliomas and are important for diagnosis and prognosis for these types of tumors, or oftentimes the driver mutations. And so those were our findings there. And finally, we wanted to do was take together all these different predictors of glioma distribution and see, are they explaining the same variants or different variants in glioma frequency? To what extent are these interdependent versus independent? And so PLS1 and PLS2, those refer to our genetic factors, or genetic categories there. And we find that each of these factors, the connectomic, the cellular, and the genomic factors, are independent of one another, and altogether they explain 58% of the variance in glioma frequency, which is a pretty high number. We know more things are involved, but we've found that this gave us a good sense of the multifaceted variables that are important for demonstrating what regions are more vulnerable to oncogenesis. And so our takeaways here is that gliomas appear near the brain hubs. They localize to regions populated by their cells of origin. And we know the genetic factors also influence their localization. Now, finally, what we wanted to do, we have these findings that there's all these independent things that seem to be related to oncogenesis. We wanted to consolidate this into some theory that explains everything that we're reviewing. And how we started to tackle that question, which would involve explaining the migration patterns ultimately of these tumors. We really honed down on this notion of subventricular origins of gliomas. So there was a recent Nature paper that was very interesting, where taking this idea that gliomas might be originating from neural stem cells in the subventricular zone, they biopsied both the subventricular zone and the tumor for a subset of patients with glioblastomas. And they found that driver mutations in the subventricular zone that were present at higher levels in the GBM itself, suggesting that the tumors had originated here in the subventricular zone and migrated elsewhere to form a massive tumor. And so we were interested in testing this idea, because if you take it seriously, this would suggest that the location patterns of gliomas would be very strongly clustered across each horn of the lateral ventricles. And so to do that, to take this neuroimaging type of analysis and to test this subventricular zone hypothesis, we took lesion masks from the Cancer Genome Atlas, and we took those masks and turned it into this huge matrix that you see here that goes voxels by patients. And we implemented an algorithm called independent component analysis to take that big data and decompose it into 3 different components that explain most of the variance in terms of lesion locations. We're interested in what these maps would look like, because they would ultimately identify clusters of gliomas, and to what extent do they relate to the lateral ventricles and large-scale brain networks was our main question of interest here. And so when we do that, when we find these clusters of lesion location, which we call lesion covariance networks, we find that each cluster coincided with a distinct horn of the lateral ventricles. So you see that this purple one here coincided with the frontal horn of the lateral ventricles. The green one coincided with the posterior horn of the lateral ventricles. The blue one here coincided with the inferior horn of the lateral ventricles, suggesting that the tumors might be different. That what explains the heterogeneity of tumor location is just origins from each different horn of the lateral ventricles. Now, the other thing we wanted to know is whether or not a lesion following in one or another of these different clusters was this associated with any genetic phenotypes that we or genotypes that we maps onto interesting biology that could ultimately be targeted. So we did differential gene expression across these different lesion locations and found that there's this neuronal to immunological axis that these lesion locations tend to map onto, where LCN1, which is where the more frontal tumors were located, those types of tumors tend to express genes involved in synapse formation and neurological processes in general, whereas you get more immunological gene expression in the tumors that appear more in the parietal lobe and the temporal lobe, here with T cell proliferation and immunological signaling showing up as differentially expressed genes in those categories. And finally, we're interested in looking at the extent to which these clusters that we identified, to what extent do they significantly coincide with the locations of large-scale brain networks. And for each lesion covariance network, we found that there were multiple networks that corresponded with them, even after controlling for confounding spatial autocorrelation and for multiple comparisons. But I think, like, visually here, it's striking the extent to which you can see the correspondence between some of these networks and where the tumors tend to appear. So here with the anterior thalamic radiation, this follows quite nicely our cluster of lesion location sort of derived from just data-driven analysis. So it was the anterior thalamic radiation for LCN1. Posterior default mode network was most associated with lesion location for LCN2, and it was an unsniffed fasciculus for this cluster of lesions appearing near the inferior horn. So taking all of this together, I presented lots of different work highlighting 3 distinct findings. One, that these diffuse gliomas participate in large-scale cognitive circuits. 2, that their location patterns relate to brain networks and neural stem cells. And finally, with this last study, we're really honing in on this hypothesis that these tumors are beginning in the subventricular zone, and network-based spread from the subventricular zone to the cortex is what explains these lesion locations ultimately. Now, our future directions, things that are still unclear and things that we're still actively investigating, is whether there's a difference in this story if we consider IDH mutated versus IDH wild type gliomas. There's most of the research on subventricular zone origins focused on high-grade gliomas. It might be that there's a different story for low-grade gliomas. That's something that is certainly a possibility that we'll explore further. Then really what we are interested in is whether and how we can target these spreading patterns clinically. Does that mean targeting radiation or surgery in regions where we expect tumors to migrate next? Can that result in better clinical outcomes for our patients? Is certainly a question of considerable interest. And finally, in terms of the interaction between this and other approaches towards curing gliomas, our vision here is that maybe by slowing down the tumor, stopping it from migrating and progressing as fast as it is, it's referenced by the Red Queen hypothesis we talked about in the beginning. Can that allow our other systemic therapies to catch up and really defeat this tumor? That's ultimately what we're interested in and want to see. And what I think the vision is for how this is going to really make a difference for how we are thinking about gliomas and how to best manage them. So I've had a range of different collaborators from the University of Cambridge and the University of Pennsylvania have been really crucial for pushing this work together. I just want to thank all of them, and most importantly, thank patients who, voluntarily, decided to participate in these studies and are contributing to our growing understanding of the pathogenesis of gliomas. And thank you all for listening.